So let's talk about some things that can affect enzyme function. Very important because, as you know, um, cells have to maintain something that you might remember from freshman bio called homeostasis, which means that we have to control when these enzymes are working and when they aren't. Cells need to know when to divide, when to move, when to react to some sort of stimulus. So, in order to do all that, they have to have things to control their function. One of the things that controls enzyme function is the concentration of enzyme. Um, in other words, the more enzyme you have, the faster the reaction, the higher your rate of reaction is going to be, right? Like, ooh, that's a nice looking graph for a freehand. That's not bad. Of course, it starts to approach its limit as you start to run out of substrate, right? If you continually added substrate, you could maintain a somewhat high rate, but eventually this will slow down and drop off once there's no more substrate available. So this is what happens when you have a higher enzyme concentration. But higher constant is equal to higher output rate. Okay. Okay. So the next one, this might naturally be where your mind goes after this, is substrate concentration. What happens to the substrate concentration? Well, as you have an increase in substrate, so too does your substrate concentration go up. Right? I only do that below so you can see it. It has the relationship between those two lines is simply that they both increase. So the higher the concentration of substrate, higher rate, right? Of course, again, as you start to run out of substrate over here, you are going to start to, this reaction is going to start to slow down. One of the major things I want you to get to remember about enzymes is that enzymes are not used up in chemical reactions. So the only thing that does get used up is substrate. So I'll write this over here as well. I'll write this down here. Enzyme is not used up in reactions. If you add more substrate, that enzyme will continue operating as normal. Okay, so let's go ahead and move on to the next thing that we'll look. Okay. Oh, spoiler right there. Temperature, another thing that can affect enzyme function. Sometimes, and we'll pretend that this x-axis over here, in this case, is increasing based on temperature. Lower temperatures are over here. Higher temperatures are up here. Enzymes are going to have some sort of optimal temperature, right? Some sort of temperature at which they operate really nicely, right? And they actually enjoy this temperature. Maybe it's 29 degrees Celsius up here, right? And it just made that up. You'll notice that this function kind of drops off and goes to zero beyond a certain point. This is because our enzyme will denature past this point. If it gets too hot, all those hydrogen bonds that are holding that tertiary structure of your enzyme in shape will break down and the enzyme will denature. And of course, below, where it's too cold, there's not enough energy in these molecules. They're too slow, right? So there's not enough energy in these enzymes to enable some sort of reaction. So too low in energy. Too low in energy for reaction to take place. So there's always kind of like this Goldilocks zone of temperature. Likewise, pH. Okay. Likewise, pH kind of operates in a similar way to temperature, in which you have an optimal pH, in which on either side of this, if it's way too basic for this enzyme to operate, it will denature, right? If it's too acidic, it will also denature, right? Meaning that there's an optimal pH. Let's call it a pH neutral, which is 7. So pH 7 up here. This enzyme would operate best at pH 7, right? And this really depends where these enzymes are active in the organism that they are functioning in. For instance, if you are a thermophilic bacteria that lives in hot springs, you're gonna be active at a very high temperature. and whatever pH your surrounding environment is. The bacteria in your stomach, uh, they would all have enzymes that operate at pH 7. You have enzymes in your system that operate, I'm sorry, not pH 7, but more acidic pH, down in the 2 range, 1 to 2 range. You would have enzymes that were more active down there in your stomach, right? So there's different optimal pHs that enzymes can operate at. There's different optimal temperatures that enzymes can operate at. So both of these, temperature and pH, there is some sort of optimal level. Salinity, I'll just talk about this one briefly, is very similar to pH and temperature in the sense that there's optimal salinity. If things get too high in salt, life is just not possible. It's impossible to keep any sort of moisture in the system. If you can think of the Dead Sea, nothing really living in the Dead Sea. There's a reason it's called that. It's because it's too saline. There's too much salt in there. Activators are things that activate enzyme. I'll talk about those a little bit on the next slide. And inhibitors are things that inhibit enzyme action. Inhibit means slow down or to stop, right? Okay, so let's move on. Compounds with shell bang enzymes. I'm not going to talk too long about this because we'll have plenty of time to talk about this when we talk about when we get further on in this unit. So, activators. Some of these are called cofactors. These are small inorganic compounds and ions, such as iron and hemoglobin. You guys may know that, may or may not know this, especially if you're in the medical pathway, but hemoglobin transports oxygen in our blood, right? And it needs iron in order to operate. Without iron, it can't operate. This is a cofactor for hemoglobin. This is something that helps it operate. They are going to be typically bound to some region of these proteins. Coenzymes. These are non-protein organic molecules that activate, and they can operate. This is a very simplistic diagram, but I kind of like it. You don't need to know these words down at the bottom, holoenzyme and all that. You just need to kind of understand that coenzymes are something that can exist. They are something that kind of comes together with an enzyme in order to allow a substrate to fit. So this cofactor here, coenzyme, can fit into a spot in order to make the shape correct for the substrate. So this keeps this enzyme from being active when you don't want it active, right? Which there's many cases in which that might be the case. They can bind temporarily or permanently to the enzyme near the active site. Many vitamins operate like this. Compounds which inhibit enzymes. Inhibitors. These reduce enzyme activity. They keep enzymes from working. And when we talk more deeply this year about gene expression, we're going to talk about some of these inhibitions more specifically. The first one I want to talk about is competitive inhibition. This is when a inhibitor competes. If you look at this competitive inhibition example right here, this competitive inhibitor is competing with the substrate. Look here at the substrate. It's competing for this active site, right? Okay, keep those words in mind. Active site, substrate. And this competitive inhibitor is competing with that. Non-competitive inhibition operates when you have an inhibitor which latches itself on somewhere away from the active site, but it changes the shape or conformation of the active site, which leaves the which leaves the substrate unable to bind to this enzyme, right? So this thing cannot bind right here. Similarly, this substrate cannot bind over here, right? Irreversible inhibition is just inhibition which can be competitive or non-competitive, but it can't be reversed. Feedback inhibition is kind of a very specific case where you have, let's say you have, I'll draw a little metabolic pathway here. You have molecule A, right? Trying to get a molecule B, get a molecule C, right? And let's say that these are catalyzed by enzyme 1 and enzyme 2, right? Well, let's say once you've made a bunch of C's, so that this enzyme's been working and this enzyme's been working, and you've made a ton of C. Well, eventually this C can act as an inhibitor to this enzyme 1, right? So it can come in and actually either compete or it can non-compete down here. But it can inhibit this enzyme 1 working. If it inhibits enzyme 1, that means that C can't be made anymore, right? Now, what's nice about this is it lets the cell or organism at some level know that, hey, we don't need to make any more of this because we have plenty of it. It's going to come back and inhibit it automatically, and you won't make any more until you deplete it. And once you deplete it, it could detach, right? And these enzymes will be freed up, and then all of a sudden, this process will start all over again. That is feedback inhibition. Okay, so that's it for environmental impacts of enzyme function. I will see you next in topic 3.4.